So now we are going to talk about the prokaryotic DNA replication. And for that, we have to know that in both eukaryotic and prokaryotic DNA replications, steps are the same, but there are subtle differences that I'm going to talk about. But for the uniformity of this lecture, I'm going to first detail the prokaryotic DNA replication, and then I'm going to talk about a eukaryotic one to tell you the differences. So the first thing to note is that the prokaryotic DNA replication is semi-conservative. Now, what does semi-conservative means is that we have a DNA molecule. The DNA molecule is going to separate, and then a two new strands will be formed simultaneously. So the same molecule separated, and then daughter strands were formed of those separated DNA molecule. So that is a semi-conservative model of DNA replication. Pretty simple. Now we talk about prokaryotic DNA replication into three steps. One is initiation, elongation, and then termination. We are going to start with initiation. So with initiation, we have a DNA molecule with us, okay? But what is important is that we need a part of this DNA to unwind. And that happens at specific locations. And those specific locations are called as ori, or the origin of replication. And the origin of replications are basically consensus sequences, which means that they have the same order of sequence at each site. And they are always AT-rich segments. Why they are AT-rich segments? That's important, a very good question if you ask us, or me actually, is that A and T are double bonds. They, rather than C that has triple bonds with hydrogen bonds with guanine. That's hard. You see, that's hard to break, but it's easy to break two bonds, right? So that's why for melting or separation of strands or breaking of bonds, that's in case of heat or whatever, and that happens in AT-rich segment. That's easy to break. In case of prokaryotes, we have DNA A proteins. These DNA A proteins are going to so these DNA A proteins are going to, they actually bind to the AT-rich segments and they melt. So this is a DNA protein. It's going to bind to an AT-rich segment like here, and it is going to break those bonds. And breaking of those bonds will form a replication fork. That forms a replication fork. So I'll tell you one more thing here, is that this is your DNA molecule. It's like this, right? And a DNA A protein is going to act on it. So DNA A protein is going to act on it, and it is going to form something like this. So everywhere else there are going to be bonds, but the bonds are broken here. This right here is called as the replication bubble. But if we cleave it and we see only half of it, we see this is the replication fork right here. Okay, that said, that was some easy concepts to understand. Now we're gonna get into the nitty-gritty stuff. Now, of course, it's not easy to just form a replication fork. There is going to be something that breaks these bonds. So we have definitely melted a small region. So this melts only a small region of AT-rich segment. There is another enzyme called as a DNA helicase that is going to cause unwinding of the DNA. So it is going to unwind the DNA more and more and open the DNA up more and more. That's called the DNA helicase. And in prokaryotes, it's DNA B. And DNA C is another protein that binds DNA B to DNA. Okay. Then there is also another protein that's called as a single-stranded DNA binding protein. These are not enzymes. Before, what I talked about DNA helicases and DNA A protein, those were enzymes. But single-stranded DNA is not an enzyme. It is just a protein. And what it does is that it shifts the equilibrium. So we know that this is double-stranded DNA. It is going to form single-stranded DNA, but the process is reversible because those bases that we opened up, they can bind back together because of attraction, laws of attraction, right? So in order to combat that problem, what nature has done is that it has these single-stranded DNA binding proteins. It is going to keep these bases in place and prevent them from binding with each other again. So it shifts the equilibrium from double-stranded DNA to single-stranded DNA, and it makes this reaction irreversible. Like that. Also, important to note is that their binding is cooperative. This means that if one molecule attaches here, it is going to increase the affinity of the bases to allow for more single-stranded binding proteins to come and get attached. In addition to providing a template for the polymerases, the single-stranded binding proteins also protects the DNA against nucleases. How so? Because they're binding, they're literally hugging the DNA molecule, so another incoming nuclease cannot attack the DNA molecule. And also, so these were the two problems that we solved. One was that we unbind the DNA, and then we prevented the DNA from, bonding with each other again. Now what we're going to do is that there is another problem. That problem is of supercoiling, as I've drawn here. So this is a DNA molecule, and it is opening from here. If I'm opening it from here, you see this opens here, and then there's going to supercoil here. So you can experiment it. You can take a thread at your home, and you try to, there is a, it is all coiled and all, and you try to open it with two loose end strands, and you try to stretch it. You will see it gets more and more clumped and coiled, and it won't open further. That also happens with DNA. DNA is also thread-like material. It also gets super coiled. Now, this is a big problem. In order to solve this problem, we have topoisomerases that prevent supercoils. Now, what it does is it first is going to break the strand, and then it is going to reseal the strands. So it is going to, there is a, right here you see there is a supercoil. What a topoisomerase is going to come, and it is going to cut. It's going to create a nick in the strand. And when there is a nick, which means a cut in the strand, this is going to cut like this. When this cuts, the tension releases from behind. The tension is going to release, and then the coils are going to open up, and then the DNA topoisomerase is going to reseal it back. That's what topoisomerases do. Now, there are two types of topoisomerase. Type 1 is ATP independent. It does not require ATP. But type 2 requires ATP. Easy way to remember this is that ATP has T in it and type 2 has 2, so 2 starts from T. So ATP dependent type 2. Type 2 is called DNA gyrase. Now, something special about DNA gyrase is that what it can do is in a supercoiled region, it is going to create negative coils. So it's going to create nicks, and then it is going to create negative coils that open up this tension that's here in the supercoil, and then it reseals it back. And that's a special property, but DNA gyrase is not present in humans. They're only present in prokaryotes and in plants. That is also good news for us, because now we can create drugs, and the drugs against those prokaryotes. So chemtothecins and etc. etc. There are many drugs that should be, we're going to talk about them later on against the DNA gyrase. Now, initiation is done. We are now going to do elongation. For elongation, another important thing is that we have the pre-priming complex. So this right here, this whole stuff that I talked about is called as a pre-priming complex. And now for elongation, we are going to introduce. Now, the thing about polymerases in DNA replication is that they need something to work on. It's just like archery or football. Like in football, all the footballers, they need a goal, right? They need a netting to kick the ball into so they can goal. Or in archery, they require a target to shoot an arrow to. The same is here. Polymerase needs something to work on. Like we need a notebook to write on with a pen. An empty pen is, we can't do anything with it. So it cannot on its own work on the other strand. So what we have the solution for is that we have a primase. Primase is an enzyme which is basically an RNA polymerase. And what it does, it produces, right here, if I change the color, it produces a 10 nucleotides long RNA. It's important. It produces RNA, which is 10 nucleotides, approximately. It's not hard and fast rule. 10 nucleotides. So it's important to know that it is going to contain uracil in it. And now what happens is that the RNA is, this strand is 3' to 5'. Now we have this 5' area in which now the polymerase can work on. Pretty impressive. So it is complementary and anti-parallel. And we have the primosome. Now, what is a primosome? When we have the primer, so right here, the primase, when primase enters the pre-priming complex, that is called, this whole together is called as a primosome. Okay. Now we have a primer, we have a primosome. We're now going to talk about leading and lagging strands. So the leading strand is okay. Its life is set. The leading strand is going to go on and on and on. But the problem is the lagging strand. It is 3' to 5'. So the stuff that is going to work is the 5' to 3', short segments are going to get produced. And the primer or the primase has to jump Here is going to be a primer. Here is going to be a primer. And it is going to form DNA in form of fragments. Those fragments are named after the Japanese scientists that found them, and those are called as the Okazaki fragments. Simple stuff. Now, the DNA polymerase 3, now we're going to talk about some other details of it. It has different subunits, but it has beta subunit that is going to encircle the DNA. So this is the DNA molecule. It is going to 3D encircle the DNA molecule and then move on. So it is like a sliding DNA clamp. And this is facilitated by another protein called as the clamp loader and plus ATP. The strand is going to grow in 5' to 3' direction. And then it also our little fun stuff is that there is going to be 3' to 5' exonuclease activity. So it's going to be, it goes opposite and it forms exonuclease. So it is proofreading. For proofreading in opposite direction, it is going to exonucleate it. Okay. Then we have DNA polymerase 1 as well, that has 5' to 3' exonuclease activity, and that is for removing the RNA primer, because we don't need that. We don't need uracils in DNA, of course. So important to note is that DNA pol 3, so pol 3 has 3' to 5' exonuclease activity, and pol 1 has 5' to 3' exonuclease activity. And then another enzyme is the DNA ligase. What it's going to do is it's going to form 3' to 5' phosphodiester bonds or linkages between the groups of the NICs, right, of these molecules right here, the adding of the bases, and then we have the whole molecule, the whole DNA molecule, and then it is going to join. The last step is termination, and in termination it is by the sequence specific binding of TUS. So we have TUS protein on the TER side. So TUS is the sequence, and TER is the site. TUS is the protein. I'm very sorry. TUS is a protein and TER is at the sequence site. And it stops the DNA polymerase 3. Right. And now about the eukaryotic DNA replication, the only thing is that it occurs during S phase. And the eukaryotic DNA polymerases, there are certain points about it. I'm just going to read through it. It's very simple. All the other steps are the same. It's just the polymerases are different. So we have polymerase alpha that has a primase subunit and a 5' to 3' polymerase subunit. Then we have the polymerase eta that completes the DNA synthesis of the leading strand. And the polymerase delta, it elongates the Okazaki fragments. And it has but polymerase eta has a sliding DNA clamp, right? That is called as a pCNA, proliferating cell nuclear antigen, and both can proofread. Polymerase beta is gap filling in DNA repair. We're going to detail DNA repair in upcoming lectures. And then polymerase gamma, it replicates the mitochondrial DNA. And that is it. That is it for today.